I want to talk to you about a disease I'm sure you know about, that recently discovered by both the Japanese and my good friend Steve Holland at the NIH. And it's very common in the Middle East. I'll tell you, most of our patients are coming from the Middle East because of the first cousin marriage. But I wanted to start by acknowledge something that Robert Good said, and that is these diseases, the primary immune deficiencies, are experiments of nature which teach invaluable lessons on the immune system. And actually, when I came to Boston to train in pediatrics, I had a grant from the Lebanese government to go to Bob Good. But then Charlie Janeway, who was the chairman of pediatrics there, called me 3 months later, and he was a real, he had to offer me the job. When I was a student in Beirut, I filled in 10 informational packets, and I got 10 applications, but I didn't have money except to mail one. So I mailed only one to the Children's Hospital. And Charlie Janeway sent me a very terse Western Union telegram saying, be here on July 1. And by September 1, he called me in his office and he said, if you are going back to Lebanon, you should train with the guy who took my place, Fred Rosen, who was my mentor. I said, but I'm going to Dr. Good. And he said, no, you call him. And I said, Dr. Janeway said, you stay here. Charles Janeway was with Ogden Bruton, discovered X-linked agammaglobulinemia in 1952. And he is the grandfather of pediatric immunodeficiency. And of course, Bob Good and Fred Rosen had their own big contributions. So I'm going to use the DOCK8 deficiency as an example of what you can do with the immune deficiency, how much you can learn about the immune system, because these are absolutely fascinating diseases. So it is one of the two known hyper-IgE syndrome. There is a well-known autosomal dominant form, which would be rare, relatively rare in countries with a lot of first cousin marriages, and that's due to STAT3 deficiency. And there is the autosomal recessive form, which is due to DOCK8 deficiency. And I just want briefly to talk a little about STAT3 deficiency because, as you know, it has skin abscesses, pneumatoceles, and candidiasis. And it is an autosomal dominant disease, but it also has, since STAT3 is expressed and is important for the development of many tissues, it has extra immunological manifestations. And that's the first thing, of course, you do when you look at a child with immune deficiency. You look for manifestations outside the immune system because that will put the gene you are looking for in a different category. Gene expressed only in the immune system or a gene expressed in the immune system and in other systems. So if a child has it's SCID type forms of SCID and he has also muscle weakness, you think maybe of the diseases like ORA1, calcium channel diseases, etc., because they affect everything. But if he has only infections and nothing else, then most likely it's a gene in the immune system. This is both. And they have these coarse features, candidiasis, pneumatoceles. And the problem there is that they have a mutation in one of the alleles, but it's a dominant mutation. So the protein is expressed, but it has a point mutation of STAT3, which is a transcription factor that works in the nucleus, but it's usually present in the cytoplasm. And STAT3 is activated upon binding of a cytokines, certain cytokines like IL-6 and IL-21, which are, in this case, the two examples I gave you are important for forming germinal centers in the case of IL-21 and for plasma differentiation in the case of IL-6. And those STAT3 molecules get then phosphorylated because the receptor gets together, aggregates, recruits JAK kinases that are, I lost the K somewhere, and they are kinases that phosphorylate STAT3. Then one phospho group on one of the STAT3 is recognized by a sequence on the other one, and vice versa. They make a dimer, and the dimer now can go into the nucleus and do transcription. The mutations, as I said, are dominant, so that because it's a dimer, if you have a mutant allele and a normal allele, then only 25% of the molecules will be made of two normal products, and the bioactivity is 25%. In contrast, DOCK8 deficiency wipes out protein expression. There is really very little protein expression. We saw it in only one Saudi boy who had a splice mutation, and it was decreased by about 60-70%. These individuals have not only recurrent sinopulmonary effusion, but have a classical eczema. and food allergies, as you would expect from the previous talk, and susceptibility to viral infection. They also have a very bad problem with making antibody responses. And this is how they look like. And the eczema is obviously the same as you see in any child with eczema in the antecubital area, etc. And they have these warts and molluscum contagiosum, etc. This is the structure of the protein, and the most important thing to remember, it has a domain that brings it to membrane, and a domain that works as a GTPase. It activates small GTPases like RAC and Rho. And the question is, how does DOCK expression in B cells influence antibody memory? So we collected several patients, and the study I show you is made on 10 patients, mostly from the Middle East, a number from Turkey, Kuwait, Lebanon, Syria, etc. And they all had clinical characteristic of the disease and no protein, either in their blood or in EBV line we made. And this is what happened when you look at these patients. If you look at their antibody response, we knew that 5 of them were tested before they were started on gamma globulin therapy, and 2 of the 5 responded to tetanus with a protective response, whereas normal children should be 100%. And none had responded of the 5 to Hep B or to Pneumovax, and the HIB 20%, versus virtually, except for the Pneumovax, virtually 100% in the normal individual. And they have poor anti-IgM antibody response as well. But the fascinating thing is that 2 of these children were immunized and followed before they started on gamma globulin therapy. And as you can see, they made not a bad response right after immunization. One of them made a very good response. But then when you came back a year later, it was again back to 0. And this is a booster response of their tetanus. They had been immunized as babies. They had received the triple vaccine as 2 months, 4 months, 6 months. So they are unable to sustain their antibody response. They make one, but so what's happening? This is a B cell. That catches antigen here, but in order to make an antibody, it has to either see a T cell that has CD40 ligand, and it will engage CD40. Artificially, you can do it in vitro with anti-CD40 in the presence of cytokines. Or it sees microbial DNA, such as CpG, coming from the viruses or bacteria. And the ligand here is CpG is internalized, taken into this endosome. And the endosome contains TLR9, toll receptor 9. Again, the Nobel Prize this year was given for the discoverer of TLRs. And this also gives a signal that allows the B cell to differentiate. And like so much in immunology and in life, these 2 things synergize. So you get synergy in real life. So we decided to look at these kids. This was a great opportunity. And actually, we looked at them because they have viral infection. And we thought maybe they have a problem with their interferon 1 production in response to microbial DNA. But it turns out it's not the case. They have a B cell problem. And this is seen here. Those individuals, by the way, their T cell numbers are low, but their B cell numbers are either normal or high. However, if you look at the marker for mature B cell, which is CD27, which are the marker for switched B cells, memory B cells, this marker is present on about 23% of the B cell of a normal individual, but very, very poor in these patients. So they have a lack of these CD27 positive memory B cell, and virtually all their B cells remain naive, even though they have seen multiple infection, because they don't have any memory. And if you just focus on the top 2 lines, it shows that if you look at the response to CD40, it's completely normal. There's a little depression here, but that has to do with the fact that they don't have memory B cells, and naive B cells are a little poorer in responding to this ligand. But if they respond, if you try to stimulate them with bacterial DNA, which in normal B cell gives you very good proliferation, IgM and IgG secretion, it's horrible. And if you look at this receptor, TLR9, for microbial DNA, it uses an adapter protein called MyD88. It allows B cell to proliferate, mature, become memory B cell and plasma cell. And both the receptor, TLR9, and its adapter protein, MyD88, have a role in antibody response. It's actually, there is a big push to use it as an adjuvant, and MyD88, if you remove it from B cells or from mice, you get a very poor antibody response. But there is nothing wrong with the expression of TLR9 in these kids. They are completely normal. They simply don't respond to TLR9. They express normal amount of protein. And after you feed the ligand for TLR9, the localization of TLR9 changes. It moves from the endosome, then it forces the endosome to fuse with the lysosome. And the signaling is slightly different. In the endosome, you activate NF-kappa B. In the endolysosome, you tend to activate interferon type 1 production. So we looked at localization using multicolored stuff. We have one of these sophisticated machines I know nothing about. They don't even let me come close. And they look all fine. So it's not a matter of expression. Until now, and then I'm using this to illustrate how much fundamental immunology we can learn from one genetic defect. Until now, there are two pathways of TLR9 signaling. Both of them use MyD88. One uses IRAC. We have the patients deficient in MyD. Jean-Laurent Casanova has described MyD8 deficiency. There are patients with IRAC4 deficiency. And this goes to activation of the transcription factor NF-kappa B, which gives you the inflammatory mediator that you see after an infection. IL-1, IL-6, etc., that come from monocytes and DCs. And also NF-kappa B is important for B-cell activation and for expression of AAICDA, which is the enzyme that allows B-cell to switch and which also causes a primary immune deficiency, one of the hyper-IgM syndrome with only B-cell manifestation because it's expressed only on B-cell. Or it activates another pathway that culminates in transcription factor IRF7, and that drives type 1 interferon production that's antiviral. So we decided maybe the DOCK8 is present. I mean, there is so much not known about this pathway. Maybe the DOCK8 is one somewhere in these two pathways. So we looked for both direct activation of NF-kappa B and the production of inflammatory cytokine and of some B-cell activation marker. And independently we looked, and that's what we started really thinking maybe there is a problem because of the viral infection in type 1 interferon production. So we looked at peripheral blood from the real patients also and looked and see if there's interferon production. And to our surprise, without going to detail, everything is normal. Make IL-6, make interferon, activates NF-kappa B. So this is not what the problem is. As I said before, one of the domain of this protein activates small GTPases by taking GDP out and putting GTP in. And we thought that, well, maybe that's the problem. And somehow this is entering into the TLR9 pathway. But that was not the case either, because the GTP activation in the normal and the patient look exactly the same. So that was not the issue. So now we started thinking maybe there is another pathway going on. Now, knowing what we know from the hyper-IgE syndrome patient and from the biology of STAT3 and its importance in driving the program for B-cell memory and driving B-cell differentiation into plasma cell, we looked at STAT3 activation. And without going into the detail, you can clearly see these are purified B-cell that if you put CpG, you phosphorylate STAT3, and if you put CpG on the patient, you don't. And the data is here. And this is not because they have only naive B-cell, because naive B-cell respond very well by phosphorylating STAT3. So somehow, the CpG ability the bacterial DNA ability to phosphorylate STAT3 is disrupted, but without disrupting, like in the dominant STAT3 deficiency, where you don't have really good activation, the activation of STAT3, if you use IL-6 or IL-21, is perfectly okay. So this is specific to the activation of B-cell by bacterial ligand through that pathway. So now we have the first link. And CpG is not known to activate the JAK kinases and anything. So it turns out that, and then we said, well, maybe STAT3 is the problem. And yes, it was, because now we went back to our collaborators who have all these STAT3 patients, and we got them from all over the world. And they have 25% activity, or some of them a little more. And they were very deficient in their ability to respond to CpG. But they responded to CD40 and IL-4. So yes, this is the problem. So now how do you connect bacterial DNA receptor TLR9 to STAT3 and DOCK8 somewhere in the middle? So how is this? So how is this happen? What is this? I messed a lot somewhere. Oh, yes, okay. I am going to be on time. I took a lot of details in the morning and I forgot that. So without showing the biochemistry, because it will be terribly boring if you are not really a biochemist, and so this is what's happening. This disease allowed us to discover the following pathway, which is totally unknown, and the paper is coming very soon. So we took various approaches and showed that what happens is that there is a complex, preformed complex, that gets even stabilized when it is recruited to TLR9. The preformed complex contains MyD88, a tyrosine kinase called PIK2, and DOCK8. And these proteins interact one by one. That is, if you take a DOCK8 deficient patient, the MyD88 is still interacting with the PIK2. And if we take a MyD88 deficient patient, and Jean-Laurent was kind in giving us one of the patients that had no protein, the PIK2 interacts with the DOCK8. We don't have a PIK2, but we did a PIK2 knockdown, and the DOCK8 interacts with MyD88. So there is a preformed complex, trimolecular complex, which then gets recruited the way this complex gets recruited to TLR9 when the ligand comes in. Changes a conformational change in the tail of TLR9. It starts recruiting MyD88 containing complexes. And this complex is totally distinct from this complex, although IRAK4 plays a role here because it's important for the recruitment of the complex. So you can see that IRAK4 is not on the slide, plays a role in the recruitment of that complex. And the amazing thing, to make sure that we are correct, is that DOCK8 has a stretch of amino acid, about 10 amino acid long. Within the stretch, it has a tyrosine residue surrounded by amino acids that make it a perfect target sequence for PIK2. And that entire stretch of about 10, 12 amino acids, once it is phosphorylated, is a perfect fit for the domain in the kinase, family of kinase called Src kinases. The major one in B cell is called Lin, that allows it now to be recruited. So once DOCK8 is phosphorylated, and we can show that now with a phosphor-specific antibody, it recruits this major B cell protein that can also be recruited to the B cell receptor in B cell and can be recruited to the IgE high affinity receptor in mast cell. And this now phosphorylates another protein called SYK, the correspondent of which is ZAP70, which causes a T cell immunodeficiency in humans. And SYK gives an alternative way of activating STAT3. So as I told you before, receptor-mediated IL-6, IL-21 activation of STAT3, which is normal in this patient, is mediated by JAK kinases. This is another pathway for activating STAT3. It depends on SYK, and we have proven all this by using multiple inhibitors and also doing knockdowns, because SYK is lethal. We don't have knockouts for SYK. So that now you have one single immune deficiency within 2 years of its discovery has allowed the discovery of something very, very important on how your B cells respond. Because in humans, you always respond in the context of something that engages your TOL receptors. Because an infection, all infectious agents have bacterial DNA and viral DNA that engages your TOL receptor. And there is volumes of data to show that if you do not engage that pathway, your B cell response is very weak and is not persistent like what it is in these kids. And that explains their lack of serologic memory. They make an antibody response, then they forget about it. Now, you tell me this is a rare disease, but I tell you right away that this is very important because the most common immunodeficiency that we see in the clinic is common variable immunodeficiency. And common variable immunodeficiency shares with DOCK8 deficiency two things. First, they don't have memory B cell, or they have very few. And actually, in that disease, the number of memory B cell is predictive of the prognosis. And second, they also have a problem remembering that they make an antibody response. So I think what we are seeing here is a really great illustration of what Bob Good said 40 years ago, is that these diseases are perfect experiment of nature, is perfect for understanding the immune system, and they give us a great opportunity to do that. I just want to acknowledge people in the lab who have done, mainly it's Haifa Jabara, who has been with me for a lot of time and Doug McDonald, who's a great protein chemist. And my previous student, who is now rejoining us in Boston, Talal Shatila, who's like me, originally from Lebanon, and he did the sequencing of these genes. He started collecting some of those patients from all over the world, and between these patients and the STAT3 deficient patients, we have lined out a wonderful array of investigators and collaborators without who really we couldn't have done what we have done, because getting this blood on FedEx and having it reach Boston within 48 hours is a feat. And some of these individuals went to the FedEx office and put it themselves in the box and got it done. Thank you very much.